So we're in session 7, measuring and enabling connectivity. My name's Graham Underwood. I'm from the University of Essex. I'm a marine and coastal ecologist. It's really great to be here, see a lot of old friends and new faces and some really great stuff today. And this session is really about how we can think about measuring this elusive connectivity at a seascape scale. It's probably easy to think about it habitat by habitat, but we're here thinking about seascapes. So we've got some interesting talks this afternoon. So we've got 4 talks and then a mystery slide at the end. So I'll tell you about the mystery slide to keep yourselves awake before the tea break. So we're going to have 4 talks and then a mystery slide. So the first of our speakers today is Dr. Maria Vozzo, and she's going to talk about identifying spatial dependencies of cross-habitat facilitation processes to achieve at scale seascape restoration.
Thank you for that, and thank you to the organizers and ZSL for inviting me today and welcoming me. I'm traveling with my almost 6-month-old, so it was pretty exciting to come here, but I appreciate the warm welcome. And I'm from CSIRO based in Brisbane, Australia, and as Elisa did this morning, I want to just acknowledge the traditional custodians of the lands on which I live and work in Australia, which is the lands of the Agra and Turrbal people, and just acknowledge that many traditional custodians have managed seascapes for millennia, and so it's really kind of coming back to that knowledge that they've had for many years that we've seemed to have lost and are trying to get back to. So happy to chat more about some of the work I've done in that area as well, but today I'm talking about some work I've been doing, as my talk suggests, identifying the spatial dependencies of cross-habitat processes and how we can use those in seascape restoration. So there is, we have some reports that have shown there's been a steady increase in seascape or multi-habitat restoration projects that have been funded. This was a report from 2022. I'm sure in the past 3 years we'd see a much greater increase in multi-habitat restoration projects. And I just want to point out in this talk, I'll be using probably multi-habitat and seascape interchangeably, but as we heard the great definition from Joe, that makes sense with the work they're about to publish as well. So this is really great news. We want to move towards more multi-habitat restoration. We've heard a lot here today, the past 2 days, and we know a lot of great examples of how large-scale and successful single habitat restoration can be, but we want to, of course, scale up to have greater impact and to restore whole seascapes. This is very natural. We think about marine habitats, we see them co-occurring. Here you have seagrass right next to mangroves. So we know that, habitats don't exist in isolation, so we need to be thinking about how we can restore them simultaneously, and also how they might be interacting in these seascapes. So I'm going to focus on positive or facilitative interactions among coastal marine habitats. So these are things like wave attenuation by reefs that then create calm water for things like seagrass to grow, or sediment trapping by things like mangroves and seagrass beds that then create clear water for corals to thrive. So there's been a lot of work by other researchers that have shown that there's really great potential for these processes to occur at across really large areas. And so in the restoration space, there's been a lot of interest in seeing, well, how can we actually use facilitation to then improve restoration outcomes? And there's some examples of people talking about this in oyster reef restoration, wetland restoration, and again, thinking about some of these longer distances over which facilitation might be occurring in restoration. But one key question that keeps coming up in these papers is really this question of spacing. Like, how far apart do we need to be thinking about these interactions? And I guess how does that match with the scales at which we're trying to restore these habitats? And just to illustrate this very simplistically, which you probably all are kind of thinking about, if we have things too close, you might have competition for resources. You also might have, the slide's a bit off, I think, but you also might actually have things like permitting barriers. So you can't restore things too close to each other because there might be different jurisdictions that are, regulating restoration of certain habitats differently. If you have things that are too far, they're going to be isolated without cross-habitat interactions, so potentially missing out on these positive interactions that we want to actually harness in restoration. And then you might get into the whole rabbit hole of, dealing with multiple agencies trying to do a seascape restoration project, which can ultimately slow down the progress that you're trying to make. But again, I guess the optimal spacing we want to see is where you have these connected systems, you have cross-habitat interactions that are positive and working together to improve the overall success of the restored site. So things like habitat connectivity, nutrient cycling, improved water quality, and wave attenuation. And ideally, this permitting is a kind of an underlying theme I'll mention a bit later. You're going to have permitting that's working together to actually be able to achieve your seascape restoration. So this key gap that I'm really trying to address is what is the optimal spacing we need to use in order to get the desired outcomes we want. So this is a paper that I worked on with a few colleagues back in 2023. It's a forward-looking opinion about how we really need to be thinking across these multiple habitats and to really help answer this question of scale and how we can use multi-habitat restoration to help us scale up our ultimate goal of meeting UN Decade on Ecosystem Restoration objectives. So in this work, I looked at, I'll just briefly introduce, so I looked at, these 6 broad categories of coastal marine habitats, so shellfish fisheries, mangrove forests, seagrass beds, coral reefs, salt marshes, and macroalgae forests. And so I wanted to first understand, well, what do we know about how these habitats are interacting across what spatial scales? And I guess at the time, there wasn't a lot of multi-habitat restoration happening, at least in the area, the people I was talking to. So I also wanted to know, well, what are some examples of where these habitats have been restored simultaneously? And so just quickly to that question, in this literature review, I found over 2,100 studies of coastal marine restoration. Only 6 of them actually talked about restoring habitat simultaneously. So again, it shows it's a really growing field, and there's a lot of questions we need to answer if we want to really scale up multi-habitat restoration. But of the ones that looked at, so then looking at the pairs of interactions between these pairs of habitats, I found almost 1,200 studies that I looked at to see, well, what are the spatial scales over which these interactions are occurring, and what are ones that they've documented. So for each of the habitats, I'll just start with these 2 just to illustrate what I'll be doing for the rest of the slide. So for each habitat type, there's a pie chart of the different facilitative processes that were identified in the literature. The different colors represent different processes, for that particular habitat type, not the relative importance of that process. So for seagrass, we found 7 different processes occurring for those habitats. And so these are examples of cross-habitat facilitation, so where one mechanism actually benefits the other habitat. So we look at all 6 different habitat types. You can see that there's quite a different types of processes reported for the 6 different habitats. So for example, corals, there's a lot of evidence of them attenuating waves, which is benefiting other habitats. And then if you look at, so then the next thing I've done is I've shown the arrow. So the arrows point to the positive interaction from one habitat to the other. So for example, shellfish to seagrass and seagrass to shellfish, there's evidence of positive interactions occurring between both of them. And if you look at the whole network, we can see the relative, the weight of the arrows represent the relative number of studies reported for those 2 pairs of habitats. So just a caveat, this represents a lot of how we've studied these habitats, not necessarily the only interactions that are occurring. So for example, in the US, there was a lot of research on interactions between salt marsh and shellfish, so that kind of indicates why there's really thick arrows there. That's not to say there's not other interactions occurring, it's just there's a lot of work that we've done in those areas, which is why it's kind of biased towards that. The interesting thing though is that of these 200 examples I found, over 85% were described on scales less than 1 meter. So that's kind of really looking at like the fine scale interactions occurring and really nested or close assemblages, which is really important and really necessary for sustaining these multi-species ecosystems. But when we're thinking again about multi-habitat restoration, we're thinking at scales, tens of meters to hundreds of meters away from each other. So there's still this kind of knowledge gap that we identified. And if you think about some of the examples we can just maybe imagine, or examples from modeling and other studies, we know that facilitative interactions are occurring on much larger scales. So for example, for wave attenuation, there's been some really great papers demonstrating how coral reefs will break wave energy, approximately 500 meters to a kilometer away from seagrass beds that then create these thriving seagrass beds. But if you think about the scale of, say, the Great Barrier Reef, that's occurring on much larger scales. So the wave attenuation from the reef is creating calmer water that's allowing all these habitats to, to persist behind it. For habitat connectivity, we know that, some juvenile fish will utilize mangroves and move out to corals and graze competitive algae, but actually, and that's been documented on the scales, again, of 2 kilometers or so, but if you think about how far some fish will migrate, this could be occurring on much, much larger scales. So that left this really big knowledge gap that we still wanted to address, and we didn't find it in the literature, so that led me to think about, well, what's a different way we could actually go about trying to kind of answer this question about this larger gap in the potential for facilitative interactions. And so to do that, I brought together a group of different experts in different biogeochemical, physical, and ecological processes that underpin cross-habitat facilitation. And so we did this through a week-long workshop, and of course many iterations and discussions after that, to identify what were some of those spatial dependencies that might have been much larger than we could find in literature, but just hadn't been quite documented the same way for us to find them in the first review. And a really important part of this work was including the practitioner perspective. So we found, I'll talk about it in the next few slides, even amongst the scientists in the room, those of us working, say, in the ecological side versus the physical side, we had very different terminology to each other to describe very similar things. And so we felt it was really important in this paper to then make sure we had a kind of a high-level summary of the different processes we were talking about so that they were really accessible to those working in the field of restoration, and we weren't, getting bogged down in too much jargon in describing what we meant and how we could actually harness these processes. So for this, we focused on just 6 different facilitative processes, for these 6 same broad categories of coastal marine habitats. Those were wave attenuation, sediment dynamics, nutrient processing, water filtration, carbon dynamics and alkalinity export, and animal and plant-mediated processes. And I just want to point out for the animal and plant ones, all of these are actually mediated by animal and plants because those are the habitat types, but this is more of the direct movement of animals and plants. So I'll give some examples in just a minute. So this is a lot of text. I don't really want to spend a lot of time on it. It's all in the paper there if you want to read more about it. But just again, the goal of this was to kind of have high-level summaries of what we mean by each of these. What are some of the key parameters by which they're influenced? So, for example, wave attenuation is influenced by, or the wave attenuation properties of a habitat is influenced by wave height, wave direction, and wavelength. And we also then highlighted the role of coastal habitats in that process. So, for example, again, for waves, like a coral reef might modify water depth and rugosity to seafloor that then, through their structure, that then is what dampens the wave energy. So we did this again for the 3 other habitats, processes. Again, I don't want to spend too much time on that, but and just to point out again, the animal and plant-mediated facilitation is really about the movement of animals. So for example, how fish might migrate between habitats or how macroalgae fronds will disturb the surface that then allows shellfish to settle. But what we really wanted to get at was, again, this question of spacing and scale. And so if there's any trade-offs or synergies in the optimal spacing of restoration sites, if you're trying to, say, maximize wave attenuation, is that then going to mean that you can't actually, you might not be able to maximize, say, water filtration? So this was where we brought together our experts and, pooled our knowledge of these different studies. So some of it was gray literature, a lot of it was published literature. The database is in the supplemental material of this paper. So again, what are the spatial scales over which these processes are occurring? The solid line we're pretty confident it's occurring at that distance. The dashed line means we're a bit uncertain, or it might depend on a lot of environmental factors at that site. So if we bring them all together, you can see that generally speaking, we have quite a range of spacing over which these different processes occur. So for example, wave energy attenuation tends to be at a larger, I could point in here actually, tends to occur, at larger spatial scales than, say, some of these more, direct modification of environment by by animal and plants providing habitat. And if we look at the the different categories of spacing here, we see that from 1 to 100 meters, we have a lot of the different processes overlapping. So that's kind of good news, because that's generally scales over which we're thinking about restoring multiple habitats together. So just one thing that came up in this conversation, though, was that, the practitioners kind of said, that's great that we have these things, the 1 to 100 meters is our kind of optimal range to, expect that we might get 6 processes to kind of overlap. But actually, their experience, they found that there's a lot of regulatory considerations that prevent restoration occurring too close to other habitats. And that tends to occur at this about 5 to 50 meter scale. So habitats actually can't be restored that close together. So that's kind of a bummer when you think about trying to do multi-habitat restoration, because if you're trying to maximize certain interactions, you might not be allowed to, depending on the regulatory considerations of the site. And so just quickly, I want to point out some of the work that Rose Foster is doing in this area. So she's at the UQ Law School, and her and Justine Bell-James have been doing a lot of work in Australia identifying the legal pathways to restoration. Rose is here at this conference, actually, and so she's been looking at some of the multi-habitat restoration, some of the pathways and barriers in the policy for multi-habitat restoration. So if you want to talk to her more about that, you can. It's quite complex. This is just a snapshot of one of her spreadsheets. So there's a lot that's at play when you're thinking about restoring multiple habitats together. At least in Australia, it'd be really interesting to hear if it's different in other places. Thank you. So a key thing, again, was to really kind of think about how can we communicate this to practitioners wanting to think about multi-habitat or seascape restoration. So we worked on this infographic that just kind of sums up what I showed on the previous graph with the solid and dashed lines. And the aim of this is actually, so don't think about this as like a continuous seascape where all these habitats are co-occurring, because they're probably not going to co-occur. But instead what you could do is you could actually use, you could say, okay, in my system we have salt marsh, seagrass, and shellfish. And so you could actually look at those columns and kind of understand the relative role of each of the habitats in providing the process that you might be interested in maximizing with your seascape restoration project. So this first figure is for scales at 1 to 100 meters. So again, we have a lot of data in this area. As we showed in the previous graph, there's a lot of potential for overlap of processes at this spatial scale. The different outlines indicate our relative level of confidence. So the more confident we are in this process occurring, they're outlined in white. Lower confidence, they're outlined in the dashed gray. If we look at the larger spatial scales, so this is scales from 1 to 100 kilometers, we see, we don't have as much information there. That's not saying these things aren't occurring, but it's maybe just at a larger scale at which we're thinking about measuring cross-habitat interactions in seascapes. And that's not to say that, again, we shouldn't think about them, because, for example, if you're, as we know, any of the fish experts here, some fish can move very, very far. So some of these processes are going to be operating across very large different distances, and it's important to consider those when we're designing our restoration projects. So just in summary, there's a lot more in that paper that we go into some of the restoration practitioner perspectives as well. So please take a look at that or talk to me after this if you want to talk more about that. But I guess, what we found is that, there's a high chance for multiple interacting processes to occur, especially at scales from 1 to 100 meters, which is pretty great news because often our restoration projects are kind of within that size range. But one thing that's really clear is there's greater research needed on the context dependency of these processes, especially temporal. So for example, if you are restoring a new seagrass bed, it's not going to provide the same properties as a really established, healthy adult seagrass bed. So we need to think about how that might influence how these processes interact, and also then over the distances over which they will interact as well. And I just want to thank all the co-authors of this work. It was a very large group effort, the funding sources, and with that, if there's time for questions, I can take a few.
Thank you. We have 2 minutes for questions. So do we have any questions for Maria? One just over here. Thank you very much.
Thank you, Maria. It was a wonderful talk. It's Tunde Gardi, and I'm with Sound Seas in the U.S. And I wondered about distance as an approximator of connectivity without thinking about currents and the rest of the physical environment, and whether there's any way to introduce that that additional way to think about potential connectivity?
That's a really great question, and certainly that's going to be really important in actually understanding how habitats might interact in a site. So this paper was kind of a first that we had a lot of practitioners coming to us saying, well, where do we even begin with multi-habitat restoration? Like, do we place things a kilometer away? Is it 1 meter? Is it 5 meters? So this was really a first pass at kind of saying, this is the rough range at which we might want to be thinking about multi-habitat restoration. But certainly like our partners at TNC, have really kind of high resolution modeling at some of their sites and can think about more fine scale currents and things. And so there we'd really want to say, okay, this is our starting point. From there, let's use this local site data and think more specifically about how this would work at that site. So certainly it's really important and not meant to be kind of a blanket solution for this. So yeah.
Great, thank you very much. Thank you, Maria. So our second speaker is Dr. Luke Helmer, who's working in the Solent Seascape restoration project. And he's going to talk about monitoring the seascape. So, Luke.
Thank you. Thank you very much. And we've heard a lot this symposium about how we shouldn't be working in silos, and this is a project that's a really nice example of that, and it brought together people that were already working in the seascape, but very much on their own and looking at their very specific habitat. And it brought those people together, and it kind of enabled that larger scale thinking and collaboration between partnerships. So I'm going to talk to you a little bit about a bit more granularity about the project and what we're actually monitoring in the Solent. So a little bit about the task at hand, what we wanted to do when we set off. So it's right back at the beginning of the project, our monitoring framework and how we developed that, where exactly we're working, how we standardized some of our data collection and working with existing systems, how we maximize opportunities as a partnership as well, and then a few updates and examples of some of the data that we're getting from the project, and then hopefully a bit of time for questions at the end. So we started off and basically looking at this, how do you monitor at a seascape scale? And the challenge was vast. We've obviously got 10 partners, all of whom have a range of backgrounds, different skills, expertise, levels of scientific knowledge, budgets, things like that. So how do you incorporate that into the partnership to maximize what you get out of that? All of the partners are working in different geographic areas, some across the whole Solent, some very site-specific, working in terrestrial, coastal, intertidal and subtidal environments. So again, the challenges with those different things and different partners working across different indicators, and I'll run you through those in a minute. Again, some of the physical actual challenges of getting out, vessels needed, harsh, hazardous environments. I haven't put the picture of me waist deep in mud. We use scaffold boards now. But a whole range of different techniques as well to incorporate into that to really maximize our outputs. So this is right back at the beginning, Joe working with all of the project partners to develop our framework, looking at actually what we wanted to get out of this. What do you want to show? How are you actually going to store the data? What platform are you going to use? Because all the organizations have their own ways of working. What our outputs will be. How is the data being collected? Who's collecting it? Who's managing it? And so, yeah, a whole range of things that you really need to consider from the start of a project. And working with all the partners, Joe came up with working within the ELSP kind of framework as well. Our main themes are around natural capital, ecosystem services, and socio-economics as well. So you can see a whole range, 15 or so indicators across all of those different themes, sub-themes, and each of those will then have different ways that we monitor across that as well. And there's a few people taking pictures, so I'll just wait for that. But it's also important to understand that whole process as well, because we do have different organizations with different backgrounds and explaining to them. So again, we've got all of our indicators and metrics and data coming in. So that feeds into the data, and also quite important as well, the media collection as well, to incorporate that within that, for example, events like this, and really portraying that to a wide range of audiences. So yeah, getting your data, how you manage it properly and store it. But then there's also the sorting and data analysis, and it's important to understand that the analysis takes a bit longer, but actually you can talk about the work that you are doing across social media and other things without doing that full-scale, detailed analysis. And so for anyone that's not aware where the Solent is, we are on the south coast of the UK. It's an area of about 522 square kilometres. So a massive area, and all of the areas that you can see here are where we are doing active work, whether that be monitoring or actually physically putting habitat back and doing the active restoration. A nice example of that is the large blue triangles, which are the locations of all of our small fish surveys. So you can see, although it's quite a straight line as the crow flies, it's quite a long journey round in a vehicle to get to all of those sites. So it's actually kind of working that into your monitoring plan and working out who's doing it, what capacity you have, and that's really important. And the small fish survey is a really nice example with kind of cash-strapped authorities. Might not be able to continue those in the long run, but actually bringing together this partnership has meant that they can continue in all of those areas where they're not necessarily required. So really useful for us, and we have that nice historic data set that we can go back and look on. Again, we've heard about how it's really important. I think we probably need to do this at a UK, Europe kind of scale as well, looking at how we do standardise. There are methodologies out there and working within them, not trying to reinvent the wheel, but it is really important to standardise how we as a partnership collect our data with different people involved. And one of those, really important, again, with the small fish surveys and things like we heard about with the eDNA earlier, collecting that metadata to see what broader scale factors could be impacting our work, particularly with the fish surveys and things like that. If you've got a really clear day and you don't get a big haul, it's probably because they can see you coming. So it's important to factor in things like that. Having things in place to make people's life easier as well. So getting your data sheet standardized, easily printable, readily available, really important as part of a large scale project. But also being open to edit and adjust those. So our metadata actually took 5 iterations of going out, right, we've got all these people working in these environments. How do we make it work for everyone? Tick boxes for things if they're not in that environment, things like that. And keeping all of the data safe as well, really important. I'm sure many people are aware of that. Having a central repository. So yeah, we do that for our metadata, our indicator specific, and it just makes life easier. Particularly with kit lists, again, everyone that's in the field, you'll get out there and sometimes you'll have forgotten something that's quite important to actually collecting it. So having that and standardizing it for people across a broad range of backgrounds is really useful. What else have we got? Yes, and also things like the latest rainfall, it's a faff doing it while you're out there, but actually having it on a spreadsheet, a link to particularly for the Solent, when the last rainfall was, things like that, makes it easier when you're inputting it back at the desk. But it's also important to, and we've heard it many times, how expensive and challenging it is working in the marine environment. We actually need to practice what we preach and work collaboratively. So we do have shared fieldwork calendars, and we try and let each other know as far in advance as physically possible. It's not always that easy because things change with weather and tides and stuff like that. But actually being able to go out as collaboratively as possible to make the most of each trip. And that's really important as well in building internal capacity and giving other people opportunities to learn other techniques. I think that's really important as a longer term project so that people can get involved. If someone drops out and things like that, it's really nice to be able to rely on others within the partnership. Again, it's also kind of removing the need to go out and do return trips and things like that. If you can collect all the data in one go, then why not? And almost as part of the outreach as well is getting the community involved. The wildlife trusts that are involved have a really nice program at the minute, teaching the teachers, getting seagrass monitoring leads up and running to kind of take over that themselves, train up other people so that it kind of maximises the amount of work that we can collect, but also takes a bit of the pressure off of them so that they can do some of the other active work that's involved as well. I'm going to run through a few examples of some of the updates and things that we've got. There are a load of our partners actually here today, so I'm going to point out a few of those for you to chat to afterwards if you would like to as well. So in terms of monitoring the connectivity of mobile species across the Solent, we have a whole range of things that we're looking at. Again, those small fish surveys, we've got the BRUV, a really interesting talk earlier about how we go forward with that actually as well. We're looking at setting up a tagging program and using existing arrays across the Solent and South Coast as well to look at how bass and other species are utilising the habitats. And again, we have a whole EDNA program, which I'm going to cover in the next slide as well. But if anyone has any questions about the small fish surveys, Zoe's in the audience. She is leading on all of that work, but here's a few kind of examples from the results that we're seeing from the first couple of years. So again, kind of really interesting to have a deeper delve into that, and probably a slightly depressing stat looking at the trend in fish populations in the Solent. But hopefully, as we start doing more active work and passive work, reducing the pressures, we'll see a reverse in that trend. In terms of our EDNA program, so across that whole seascape, we're looking at water and sediment eDNA. So really exciting results. So we're already seeing 100 fish species within that sampling program. Both species of seahorse, native to the UK. We've got 5 species of wrasse, 7 species of goby, and a whole array of kind of threatened and critically endangered species as well. What was really nice actually recently at the Native Oyster Network meeting was the presence of critically endangered eels at every single site across the UK. And everyone having the same experience that I had the first time when you find an eel in an oyster cage and you think everything's going to be static, and then it wriggles out, and yeah, it's quite a shock. And again, Hayley's in the audience. If you want to have a chat with her, she's leading on all of that work, doing all of the analysis in-house as well. But connectivity isn't just within the environment, it's actually within the human population as well, and that's really important to consider how we do that and how we, I think it was mentioned earlier, how we integrate with a range of backgrounds and different communities. So we're looking at events like this and how we are communicating, who we're communicating with, our audience sizes, the backgrounds, the type of events. And now that we're 2.5 years in, we can probably have a look at that and see where we do some more targeted outreach and engagement with different communities that maybe we aren't engaging with that well at the moment. But so far to date, we have directly engaged with nearly 75,000 people. Just on our Seascape project social medias, it's around 630,000 people. So getting quite a reach within a relatively short period of time, which is really exciting. But what's really nice is that we're actually at the point where we don't have enough opportunities for the amount of volunteers that we have. We have so much interest from the community in getting involved. We don't physically have enough events. There aren't enough oysters to scrub, seagrass to put in. So it's a weird position to be in. But what's really nice is that we have started doing these community-led events and training those people up to go out and spread the message for us. And quite a nice statistic there, over 3,000 hours of conservation work through our volunteer program, which is amazing. But we're also looking at behaviour changes as well within the environment, so looking at anchoring and seagrass and mooring in seagrass meadows. And a few statistics here. We had, again, the Wildlife Trust leading on this. We had nearly 600 responses, which is fantastic. Some unsurprising results for the demographic of the sailing community in the Solent. But what was really interesting, particularly these that literally came in this week, was actually the lack of trust in NGOs from that sailing community, and that they take all of their information from the relevant harbour authorities, the RYA and things like that, which is really useful for us of how we move forward with engaging with them and using those platforms to build trust and actually get that information out there and make those meaningful changes. And the main things that came out of that is that we do need to raise more awareness, so working with the RYA and their programs that they've got, but also providing alternatives and not just saying, no, you can't do that there. Tailoring our message and monitoring the impact that we do have across that as well. In terms of the work that the RSPB and Natural England are doing on our nesting seabirds, Holly's in the audience as well, so if you want to have a chat about birds, grab her. But some of the things that we're monitoring are up on screen, but some really nice statistics coming out of the recent reporting. Over nearly 13,000 breeding pairs, and some nice work with the sandwich terns and ring plovers, the kind of highest recorded numbers of fledglings on record, which is really nice to see. They're also monitoring predation pressure as well, which is a big problem with rats and foxes, and how and where that's happening, and how we then target some of the actions to help prevent that. And some of the work that we've been leading on at Blue around the oyster reefs, we've been recently extended our reef in the River Hamble, so that's now 0.5 hectare. It's amazing seeing all the work in Australia and the States, but it's also really depressing when you look at the size and scale and what we're doing. But it's nice that we are now starting to scale up to this. Again, we had 140 volunteers over 3 days cleaning. Yeah, a lot of oysters, 10,545 oysters, getting those on the reef. But again, as Feline mentioned earlier, we need to scale up, and how do we do that? Where do we look? And again, monitoring a whole range of indicators across that. We're seeing some really interesting results actually on this. I've got on the next slide looking at our recruitment. So these are our modified cupels, because we're working in highly navigated channels. We've had to modify them from the big ones that you saw Danny presenting earlier, that we gave them. So we've cut them down so they don't pose any risks to navigation. But what we are seeing on our reef sites compared to our control sites where we're not doing any work is actually increased recruitment of native oysters onto those cupels. So really encouraging that even within a short period of time we're getting those oysters down. And we're seeing similar trends with the biodiversity uplifts as well. So particularly with native species comparatively with invasive species, we thought that maybe dumping 400 or 500 tons of material over that area would wipe everything out, but it's actually bounced back really quickly, which is really encouraging. Again, with our salt marsh, really annoyingly, literally after I submitted this, I got a picture of the beneficial use site in Chichester, which has had a massive recruitment of Sanicornia all over that this year, which would be really nice to update you on. But hopefully at the next Seascape, we'll have some more results on our salt marsh. But again, a whole range of monitoring across there. Unfortunately, some limitations and kind of hurdles with licensing and funding and policies and things like that, causing some issues there as well. In terms of our seagrass, again, some really encouraging results coming through that hopefully we'll be able to present a bit more. But over the last 6 months or so, a huge amount of effort from Project Seagrass and the Wildlife Trust, getting huge amounts of seed planted, and again, seeing really encouraging results there. Again, important training up those volunteers to be able to lead those surveys efficiently. The realities of the system. We as a community here and as a project we are pioneering this in the UK. No one's really done it before. We don't know what works, we don't know what doesn't. But on the whole, it is working, but it was not without its challenges. It can be also challenging in terms of capacity as well. A lot of the time, because we are reliant on tides, all of our project partners are spread across the Solent trying to get to their sites on the right state of tide. So again, working as a partnership to maximise how we can do that is really important. Again, it's kind of, simple, but being organised is really key to all of that. One of the main things that, particularly that I've learned, and as probably other project partners has learned, is actually factoring in time to gather your time zero or baseline data, and actually factoring in the time that it takes to get the licenses and permissions to do that is really important. You get all the money and then you're like, yeah, let's go. And like, well, we need to actually know what it's like before we start chucking stuff in, because that's 5 years down the line. We're going to struggle if we don't. And also making the results understandable to a wide range of audiences is really key as well. So thank you very much for listening, and I think hopefully got some time for questions.
Thank you very much, Luke. We do have a couple of minutes available for questions. So do we have a question for Luke?
So Elisa from TNC again. Luke, I think we discussed this a little bit in a social setting, but I'm really curious to know what can we learn doing our restoration projects in terms of how the Solent project is doing the data management. Do you have any nuggets of wisdom that you can share with us? What do we do? Excel spreadsheets or databases in Access or smart sheets or big third-party built platforms that are very difficult to pay for because they're expensive? What is your recommendation?
It's a really good question. It's something that we are good at, but could be a lot better at, I think. At the minute, it's all kind of done in-house on spreadsheets and things like that, and a shared SharePoint drive. So I think if we could get, again, that expertise in-house to develop a platform that is easy for people to upload data and set constant reminders, because we all do need reminding to collect data that we're not necessarily going out to collect, I think is really key. And I think that's a real barrier, actually, that kind of the paid platform can be really limited. I think I was chatting, about the one that TNC had. I thought it was amazing, and then you're like, oh, it's really expensive, and a few thousand a month is not feasible. But there's some really nice examples for the other end of that from other ELSP projects in Turkey. They've got the reverse of that, are kind of outputs and how you get that and how you sort the data. But I think the beginning bit is probably something that would be really nice to get other people's opinion on how we do that as a project.
Great, thank you very much. Thank you, Luke. Luke name-checked a number of his colleagues. He's now having to leave to go to the Maldives, so he's off. So if you have further questions about that, those name-checked colleagues on the slides, do talk to them. And we can only see the heels of Luke as he disappears off to the airport, so splendid. That's the first for me chairing a session when someone's shot out that quickly. So, but there we go. Right, our next talk is Dr. Emma Ward. She's now at the University of Bangor, but was in a previous life at the University of Portsmouth. And Emma is going to talk about mixed provenance of organic carbon in temperate intertidal seagrass sediments. Emma.
Lovely. Thank you, Graham, for that introduction. So, because the session was about measuring and enabling connectivity, I thought I'd use my time to talk about how I've used stable isotopes as a tool to consider connectivity, but primarily centered around carbon provenance and intertidal seagrass here in the UK. And yes, I mean, Graham kind of alluded to it, but I should probably mention that although I'm at Bangor now, the work I'm going to talk about was done whilst I was at the University of Portsmouth. So starting at the beginning, when we think about seagrass, they can sequester carbon in two ways. So firstly, in situ, photosynthetically. When we think about this, and for this talk, I'm going to just talk about it as autochthonous carbon. There's lots of debates about what we can consider autochthonous and allochthonous, but I'm focusing on seagrass-derived carbon being considered autochthonous. And then that second process by which seagrass can sequester carbon normally comes through sedimentation of organic matter. So that organic matter originates from outside the system. So that's why we're referring to it as allochthonous carbon, okay? And what happens is they all end up mixed in your sediments, and we're trying to decide what portion of which we've got. But why are we interested in whether it's autochthonous, allochthonous? Why do we care? But that's a lot of the time because of what, especially in my work, we're thinking about greenhouse gas emission reductions, applying it to frameworks such as, I've picked this one from the from the verified carbon standards because it applies to seagrass, and that's kind of the topic for the talk. But it's focusing on how you impart, and I've put the kind of various sections up there, but in part you're focusing on storing autochthonous carbon in your sediment. And a lot of the time you go about this by looking at your carbon stock, but then you consider a value, or you come up with estimates for a value to deduct that proportion that's allochthonous. And I guess the premise for this, or the justification for this, is because autochthonous carbon, it's the focus on that is you're not wanting to look at give credits for carbon that might have come from outside your system, so your project boundary. You want to look at the greenhouse gas mitigation is really coming from the project activity that you're undertaking. And because there's been a lot of concerns in the past about double accounting across habitats. And in this sort of framework, there's various ways that you can provide that allochthonous deduction value. So that's by undertaking your own field data, by kind of looking at literature values that exist but are perhaps relevant to the system you're studying, and I'll kind of delve into that a bit, and perhaps modelling. So I thought I'd briefly talk a little bit about stable isotopes, just in case there's people in the room that haven't worked with them. I think it then nicely sets up the rest of the talk when I start talking about fractionation or various things that are kind of devolved from working with stable isotopes. Using carbon as the example, so you have two forms of stable carbon isotope. So that's carbon-12 and carbon-13. And the main difference between them is that there's one extra neutron. This is important because processes which use carbon might discriminate between those two forms. So carbon-12 is the more abundant and ever so slightly, I'm going to say, lighter version, which can lead to different ratios of these in materials. When we think about plants, seagrass being a plant, plants undergo photosynthesis, and this is a process of fixing carbon. So you might get discrimination between these lighter and heavier isotopes, such that fractionation occurs. So this means that a plant might become depleted in the heavier form of it. A really simple way, I think, to think about stable isotopes is just to consider differences between materials. So the example on the left, you're saying your isotope value of your seagrass plant and your seagrass sediment is relatively the same. So assuming most other things in your system have values that are different, you're assuming everything in the sediment was probably seagrass derived because they're matching. If we look at the example on the right, which actually shows the values from temperate regions across the world, you have a different value for your seagrass leaf and a different value for your seagrass sediment. So another organic carbon or another process has happened that has meant that those values are different. They've moved away from each other. And that ultimately suggests that there's perhaps some allochthonous carbon in your sediment. So this was one body of work that we did that's a global meta-analysis. So it looked at, using that example of differences, seagrass sediment values and seagrass leaf values that were paired across the globe. So 500 different paired values. What you can see across all of the regions is that you get this positive difference. Generally speaking, there's always a difference between your seagrass value and your sediment value. So in nearly every instance, there is presence of allochthonous carbon. So that's perhaps, you know your allochthonous carbon's there, but what allochthonous carbon is in your system and how much of it is there? And I think that really moves into the main part of my talk, which focuses on the research I did down in the Solent. And these are some of the objectives that were across this project. So looking at the isotope ratios for the different species, for different seagrass species and the different tissues, so above and below ground. As before, comparing your seagrass value to your sediment value. Then applying a multi-proxy approach, so looking at isotopes, but also considering things like CN ratios, which can be a really nice proxy when you're working with sediment, because often your samples go through an elemental analyzer before they go through the mass spec. And then 4, looking at how we apply stable isotope mixing models. And I'll kind of go through each section and you show how I used each part of this to kind of build towards like making these models and having to make decisions about how you put them together and what might be a suitable one for the area that you're working in. And I think Richard set up really nicely yesterday that seagrass can exist in different settings. And actually, within where I worked in the Solent, we've got an example which I'm going to talk about, Farlington Marshes, which is a muddy, perhaps more estuarine considered seagrass system. And then we've got a site out on Ryde on the Isle of Wight, which is a sandy, open coastal seagrass system. And perhaps we're going to have to treat those two differently. You'll see as we go through. So how did we go about it? We started by taking sediment cores, so 3 sediment cores in each site. For this part of the work, we were only focusing on the top 5 centimetres. Those cores were also taken as part of a larger body of work, so we do use the whole core, but just for other things. On the basis that the sediment in the top is the stuff that's most likely to be recently accreted. What we do is we also collect vegetative samples alongside this, so whatever is in situ or in close proximity to the site you're working in. So this might be I said about seagrass samples, but it could be salt marsh, it could be various macroalgae. If you have the capacity, it can be things like microphyta benthos or epiphytes. So those vegetative samples tend to be freeze-dried and ground down to a powder. Your sediment tends to be acid washed and then dried and ground down to a powder as well. So you're working with them, they start looking as they are, and then eventually you end up working with powders and extracting these values from them. And we did all our mass spec work at the time up at the British Geological Survey's facilities. So starting with the seagrass, although we assessed different tissue types, species, and site, so you can see above ground material, below ground material, and those two Zostra species, you can see that the main difference actually occurred between sites. When you start thinking about moving towards those stable isotope mixing models, you have to make decisions about what level you aggregate your sources. But for us, it became apparent that it's completely appropriate to aggregate them just as a seagrass source. But it does allude to the fact, again, that we might have to treat our sites very differently. So this figure shows the isotope values, but now from our sediment. The difference from before is that you've got the isotope value versus the CN ratio. And these have been overlaid onto sort of standard ranges that you get in sort of some paleo studies for sort of typical terrestrial groupings, so C3 plants, C4 plants, and then some sort of marine ranges. The black circles are for our Farlington Marsh site, so that's the more harbour-based site, slightly more estuarine, whereas the grey triangles are from that open coastal site. I think the important thing to take from this figure is those black circles are sort of shifted more to the right. They sit between those marine source ranges and your terrestrial ranges, perhaps indicating that you're going to have to consider allochthonous sources that are split across both of those. They've ended up in a mixture in your sediment. Whereas for the ride side, it's really mainly sitting in those marine ranges. So you perhaps need to think about slightly different allochthonous sources going into that sediment mixture. So I won't go too much into this table because it's a lot of numbers on a slide, but I thought it was really nice to talk about, I suppose, sometimes the way that you group things. I won't go into the seagrass because we really already talked about that we'd aggregate it by site. We only have salt marsh at our harbour site, so that only will only appear in that model. The macroalgae is where it gets kind of interesting. It was kind of referenced a little bit earlier in that eDNA talk. For us, you can kind of see just in the ride section of the algal table, epiphytic materials had to be slotted in there. So we actually, rather than using macroalgae as a allochthonous source, we had to come up with this algal grouping and put them all together. It perhaps shows that in our system you couldn't get that resolution that you might want to perhaps discern between algal sources. Sometimes you can, sometimes you can't. That's I think the thing to kind of highlight from isotopes. And in this case we couldn't, but it didn't affect our sort of priori question that we were trying to discern seagrass-derived carbon from other allochthonous sources. So then just think about what's appropriate for the question in context. So yeah, and then a couple of the other sources we considered, we're looking at suspended particulate matter, so looking at what's directly in the water column, and then for the Farlington site, looking at sort of land usage around the site. So there was a lot of pastures and agriculture, so we were using a terrestrial isotope value, but one that reflects sort of pasture-type plants, so I think C3 plants. And I think this is where I get quite excited. This is showing you what goes into the model, and then I suppose what comes out. I've got two stable isotope mixing models, so I'll run through this one first and then the next. They both essentially are a figure with two panels. The panel at the top shows you the sources you considered, and then it has your sediment, and that's essentially your mixture. So the model looks at what various combinations of these three could have ended up with this mixture. And the output of that is it gives you an estimate of the proportion along with a margin of error. What I think is really interesting for this site, so this is our open coastal site. We only considered one tracer, purely because there wasn't very much nitrogen in the sediment that we could draw out. It would have kind of convoluted the results. And we stuck with a very simple model, so suspended particulate matter, seagrass, and it was the only in situ vegetation was algal matter. What's really interesting is that the smallest proportion of organic carbon in the sediment was actually seagrass derived. Seagrass sediment, when you put both of these together, you actually end up with 84% of it being potentially allochthonous. So this is the other site. We're applying a similar process, but it's a slightly different model. So it looks at two traces, so carbon and nitrogen, and it's a lot more complex because of the nature of the site, where it's in that estuarine setting. We knew we needed to consider both terrestrial and oceanic sort of sources. We had our in situ sources, so salt marsh, our algal contributors, and seagrass. And then we've got our suspended particulate matter and this terrestrial sort of pasture allochthonous source that we're including. In the same way as the figure before, it looks at those various combinations, how they could end up with that mixture. The really, I think, important thing to take away from it is because your mixtures are sitting nicely within what we call an isospace, it does infer that those sources could end up with that mixture. If they sat outside of this, you're clearly missing something. There's a source you've not considered. Okay, so then, yeah, the output, again, as before, it kind of looks at what proportion of them and gives you a margin of error. It's a lot more complicated. It has to consider a lot more things in this instance. But it's interesting that what still comes out of this is the seagrass-derived carbon is the smallest contributor in seagrass sediment, thank you. Which is interesting when we think back to that, splitting it. We're going to get carbon accreditation, potentially for seagrass-derived carbon, and allochthonous carbon, you're going to have to include a value that you deduct. So I think some of the key things from this study, and I think in the context that I'm talking to you about it, which is about using stable isotopes, is that it identified really clear differences between both of our sites, probably related to the dissolved inorganic carbon that's in those systems that those plants, various plants are taking up. And that led to us having to consider them very separately and look at them in different sort of scenarios. What was really helpful was using a proxy alongside that. So we used CN ratios, but you can use things like pollen in sediment, sometimes to show at what point in time it switched from a saline to a terrestrial system. So you can use other things to help you make decisions about isotopes. I think that's what's quite clear, is it's best if you try not to work with them on their own. The main takeaway from my study was that the seagrass-derived carbon wasn't the dominant source in either scenario, which is really interesting when you start thinking about those greenhouse gas mitigation policies. I always like to include this picture on the right, because when I talk about my intertidal seagrass, I'm talking about these two. So mariner, that is this sort of size, and nullti, that is, again, very hair-like and small. So it doesn't necessarily, if we had a lot of talks yesterday about how traits might confer like functions and processes. So what I've talked about today doesn't necessarily translate to this subtidal seagrass that a lot of people picture. But it's definitely delivered clear and conservative estimates of potential carbon provenance for seagrass sediments in the UK. And then, yeah, because the symposium's about thinking about tools and how you might apply these, I suppose I laid out some of the decision processes that I kind of had to go through, whether that was scientific ones about, or ecology-based ones about what should be included, what's appropriate. Some of it's also about capacity and cost. So can you collect seagrass leaves across the year? Do you have the capacity to do multiple samples and get all of those sent off for analysis? Or are you better picking older leaves because they're more likely to be an average of all the processes that have happened in that plant over a longer period of time? do you collect different tissues? Ours showed that they had to be aggregated, but that won't necessarily be the case in every system. There's definitely examples where seagrass below ground materials had to be treated as a different source because it's quite different. And then when you think about allochthonous sources, I think that's probably quite key, and this is where I think stable isotope mixing models do rely on your ability as an ecologist to think about the system you're looking at and what's most appropriate to consider. So can you collect vegetation samples in situ? That helps you know it's there and it may be contributing. Is it appropriate to use literature values? For some things, we've got really clear ranges, like some C3 and C4 plants. But again, we've seen, when I looked at that map, there's not that much geographic distance between Ryde and Farlington Marshes, but we actually had to apply very different stable isotope models to them. So that's not always true. Is there data on surrounding land usage, i.e. pastures, agriculture? There was lots of mention yesterday about, water quality and sewage. Do you have additional proxies? And I think that can be a really nice benefit when you're looking at stable isotope models. And then this is something that we weren't able to apply, diagenetic correction factors. So we're applying isotope values for living tissues to see if they're in the sediment based off the fact that they might have undergone processes and sort of degraded. And then the final slide, it just reopens it back up. It's not a statement, it's more just thinking about if we're looking at tools, looking at connectivities, it's making sure that they support policy. And this was something that came out of the last symposium. So it's how we take, habitat scale policy and look at it at a seascape scale. I'll stop there.
Really nice to see, and it's quite a complicated area, the whole, it's not as simple as, a bit like eDNA, it's often cast out there as a, straightforward method, but there's a lot of complexity you need to think about. So well done, Emma. Thank you very much. So our last talk before the mystery slide is by Georgina Chandler, who works at ZSL. Her title is on From Practice to Policy, Embedding a Landscape Seascape Approach into Decision Making. And we were all agog because it said abstract coming soon. So we don't know what's going to be said. So,
it's because my talk was coming soon.
So Georgina, over to you. Thank you very much.
So it's very nice to be here. I'm Georgina. I'm Head of Policy and Campaigns at the Zoological Society of London. So I look after our policy work, our public affairs work, and our campaigning work. So it's the murky and mysterious world that doesn't necessarily dwell at the sediment layers that you guys are used to working at. It's the kind of politicians, the decision makers, the politics. So that's a little bit about what I'm going to talk to you about today. Firstly, I want to say a huge thank you to you all for you sharing your amazing knowledge and stories over the last 2 days. It's been amazing, I think, if I'm allowed a pun, to plunge into your world, your underwater world, and immerse myself in all of the work that you've been doing. Now the part you've all been looking forward to, I'm going to talk to you about influencing policy. As you can see, working on policy doesn't make for very nice slides. So this is the world that I work in. Bills and legislation and government and decision making. So I found some more seascape appropriate imagery, and I'm going to pepper this throughout my talk just so you stay interested. Otherwise, I might lose you all in the next 5 minutes. So the first rule to remember, and something I want you to take away, is always make sure that what you're communicating is relevant to your audience. Have some seascape slides. So policy matters, and I really want to emphasize this. It shapes the operating environment we all work in. It can help or hinder our objectives and present both barriers or solutions to success. It is applied at all scales, from the global, such as the United Nations, to the local, such as local councils or the Greater London Authority, which you're probably all familiar with working in. I won't claim to be an expert in marine policy, and I'm definitely not going to present a load of solutions to you. I actually really want to hear from you what you think the policy solutions are. Instead, I'm going to run through a few things in my presentation today. First, outline some theory, some advice, and some examples of how to influence decision making. So hopefully you can take that away from here with you, and use it in your work on a day-to-day basis. Second, I'm going to present back to you some of the things that I've heard throughout the course of the last 2 days, and I'm going to ask you a few questions, and I would love to hear your thoughts and input as well, and hopefully leave you with a few questions and challenges. So context is really, really important for influencing policy and decision making. And our global and national context has changed significantly in the last 5 years, and I think it's fair to say especially in the last 12 months as well. We have the Global Biodiversity Framework with ambitious goals and targets for 2030, including a 30% protected area target, which includes seascapes in it, which, despite having worked on this since 2016, I had not realised. So yes, it's in there, amazing. The EU Restoration Law was a landmark win. It seeks to restore 20% of land and seas by 2030. We heard yesterday about the UN Decade on Restoration and the amazing progress that can be made when we come together globally and locally to push restoration initiatives forward. We also heard about the rapidly increasing interest in emphasis on finance solutions and blue carbon, which presents opportunities and risks, I think is what I took away from yesterday. And I think I'd be remiss without being trendy and mentioning that AI is altering the speed and capability of what we do on a daily basis. There are also challenges. In the UK next week, we have a comprehensive spending review coming out on Wednesday. And this will shape department budgets for the next 3 years, and it will impact all of the partners that you work with in government, and it could significantly change the amount of resource they have to do their work. The actions of the US government as well are having a significant impact on the operating environment for businesses. Yesterday I was on a call and someone described to me the concept of green hushing rather than greenwashing. So this idea that kind of corporates are taking action, but they don't really want to talk about it because they're worried about consequences. So a significantly changed context of the work that the environment in which we're working. So how do we cut through the noise and try and have policy impact? Influencing policy is not a linear process. Very rarely is the job done. It's more like a continuous cycle that shifts around that context that I've just spoken to you about. There are different tools, tactics, and interventions we can use to inform and influence each stage of the decision-making cycle. And many of them we've heard about over the last 2 days. So I'm going to take you on a little spin around the policy roulette wheel, and then I'll give you an example of where some really exciting legislation was changed. So first up, we need to identify our issues. What is the problem we're trying to solve? And build your evidence base to support your case. So that's where a lot of the data, the monitoring, the research that you've been talking about over the last couple of days really comes in. Secondly, does that problem need a policy solution or intervention? Policy isn't always the solution. I won't kind of claim that, although I like to think it is sometimes. Build what that ask is. What is the change that you want to see, and how is it going to help tackle the issue that you've identified in that first step? Agenda setting. This is building your like-minded coalitions. This is gathering champions, creating a narrative for change, using those real-world stories and scalable examples of best practice that we, again, have heard about over the last couple of days. It's really, really important here. This is the area where I tend to sit, which is more the kind of advocacy and campaign side of things. Then we're looking at influencing the decisions. So that might be legislative, it might be a political declaration or commitment. And something I want to highlight is sometimes you swing backwards and forwards between these first 4 steps because you get to the decision-making process. You might not get what you want. You sort of have to go and gather a bit more evidence. You have to maybe build your agenda a bit further, and then you can kind of get stuck a little bit on this bit. But hopefully, eventually, you win. You get the decision through. But it doesn't stop there. We actually have to implement. We actually have to do the things that will hopefully tackle the issue that we identified. And we need to continually assess whether that implementation and that intervention that we've identified is being effective. Is it having the change that we wanted to see? And if not, sometimes we have to start doing this all over again. So this is not an academic diagram. This is how my brain works. So hopefully it makes sense to some of you. When we think about campaigning, there are also different approaches we can take, and campaigns is very much a scale, and it really depends on the context that you're working in, and it depends on who you're trying to influence, and it depends on who you are as an organisation as to what the most appropriate things are for you to do as an intervention. So this can range from the more kind of scientific advisory sort of services, which are think tanks, science institutes, expert bodies, media campaigns, activism, which is generally what people tend to think of when they think of campaigning. So that's petitions, demonstrations, public demonstrations, leaflets. And then advocacy, which is again kind of where my team tends to sit slightly more, which is meeting decision makers, trying to convince them to make a change, responding to consultations, writing letters. They're actually the least favourite part of my job, but something that we have to do to make sure that our voice is heard. So a quick worked example that has everything and nothing to do with the seascape in some ways, because it's about climate change. So it's using the 2008 Climate Change Act as a bit of example for how this came about. So the mid-2000s, there was a growing body of landmark scientific reports on climate change. The Kyoto Protocol was adopted in 1997 and entered into force in 2005. So that's the context that we were working in. There was very much a shift from the global level, from the UN level, down to national governments, asking them to put climate strategies into their legislation. So when people always say to me, what's the point in these UN frameworks? They never do anything. It might take a long time, but they do trigger action. So we had the Kyoto Protocol, and then notably the Stern Review on the Economics of Climate Change was published in 2006. And this was really effective for several reasons. Firstly, it was commissioned by the UK government. So if government commissions something, they tend to kind of have to listen to the results of it and implement the results of it, or at least gives you something to hold them account for. It was the first of its kind to quantify the economic impact of climate change, and it outlined really clearly the risks of inaction and put that in a way that was really communicable and translatable to policymakers. In 2005, the UK Climate Change Bill was proposed by campaigners, and I really want to give a big shout out to Friends of the Earth for their Big Ask campaign, because they managed to get over 200,000 people to contact their MP, use celebrities, really did this agenda-setting piece to try and turn something that was quite technical, quite complicated, into something that really mattered to a large group of people. They also managed to get cross-party consensus, which meant that it wasn't a party political issue, so you didn't end up with that debate of, we believe in climate change, no, we don't believe in climate change, well, we think we should do something about it, no, we don't think we should do something about it. So getting that cross-party support was really, really important. And that Big Ask campaign eventually led to the Climate Change Act being proposed in the Queen's speech in 2006. There was another 2 years of really in-depth campaigning, advocacy, agenda building. So something I really want to emphasize is this stuff does take time. It takes quite a lot of time to build that agenda, to really get that piece through. So the Climate Change Act was agreed in 2008. So you can see from sort of 2005, when that bill was first introduced, it took 3 years in total. And that is without all of that background work that was done at the UN level to set that context up ready for that. So it can be a slow process. Then, of course, it doesn't stop there. We're still implementing the Climate Change Act. We've still got a long way to go until we meet our net zero targets. We're constantly monitoring and evaluating whether we're making progress through bodies like the Climate Change Committee. And then we're making sure that the policies are being implemented that reinforce and support what was in the Climate Change Act. So it's a really good example of how science, how agenda setting, and how decision makers can work together. It was a really good blend of those different things, and there was a really clear, actionable policy ask. But that was very much combined with years of sustained pressure and activism. So, what can you do? I've got a few tips and hints and tricks. So, how can you make similar change happen or apply advocacy approaches to your work? So, context. Policymakers are operating under time pressures and political constraints. How you communicate your science and evidence must be tailored to the current debates that are happening in government. Labour have 5 missions. Does anyone know what the 5 missions of Labour is? Shout them out if you do. No, you should, because you have to tailor all of your asks to fit into one of those 5 missions if you want it to happen in the current government context. So there's the NHS, there's healthcare. I actually can't remember them all. So there's healthcare, there's education, there's domestic security, there is economic growth, which, surprise, surprise, no one will be surprised to hear that that's one of their priorities. And there's a 5th one that I can't actually remember off the top of my head, so it's obviously very important. So know your operating environment and make sure your asks fit within that. I put some oysters in just because we hadn't seen some for a while. So what? Ask yourself this. Policymakers are outcome focused. They need to understand how the science will inform decisions and what the implications of your proposals are. So you know your context now. If you're speaking to a minister or an MP, what have they said recently in speeches, meetings or announcements? What do they care about? What motivates them? If they're working in a government department or agency, how can your work help them deliver their priorities? Everybody's trying to do a job. Have you answered that so what question? If you haven't, do it, because it's very, very important. The other thing to do is keep it simple. So there's 2 graphs here. Both of them are temperature record graphs. This is the first one. This is the second one. Which one tells you more clearly what is happening to the temperature if you were going to describe this to somebody that wasn't an expert? The one with the colours, the one with the blue and the red, the one that shows it going up, the one that is a clearer line. So it's very, very important to keep your message simple, but don't be oversimplistic. Don't dumb down your results, but it's very important to try and find a way of communicating that, again, that resonates with your audience. So that might be images, maps, league tables, traffic light systems. These are all really useful tools. I remember talking to an old colleague of mine who's the chief scientist for BirdLife, and he'd produced all these amazing figures that all had lots of arrows going in lots of different directions to them. And I said to him, I was like, could you do a happy, neutral and sad face for me? And he looked at me like I just sort of suggested something terrible. But genuinely, that's what I needed to be able to go and communicate what was good, what was bad, what was okay, that kind of red, amber, green traffic light system. And it's very good to be able to test your message as well. Can you say it in a sentence? Can you say it in a paragraph? Can you say it in a page? Any longer than that is too long to communicate what you need to a policymaker or a decision maker. It's a well-known marketing trick as well, that somebody, to be able to buy something, has to encounter your brand message 7 times before they will buy it. So don't worry about repeating your key message over and over and over and over again. You might feel a bit like a broken record. You might feel like you've been saying this for years. But actually, it's a really good thing to be able to do. And lastly, make sure you invest in and build relationships, not just within the sector, but also very importantly, outside of the sector as well. You will always need champions, and knowing your context and your audience and what messages are going to work will really help you with that. Understanding what motivates these people as well, what are the pressures on them, what are they trying to achieve in their job, and create that sense of co-ownership and trust when developing solutions. So I think a lot of you here are really, really good at that. You invite government, you invite decision makers to your roundtables to the very start of your project development, and that buys that kind of co-ownership and trust, and it helps you understand what they need to be able to do their jobs properly as well. Again, another another picture. So I just wanted to leave you with a few questions and a few thoughts, and then I'm going to try and do a little bit of audience participation, but we'll see if that works. So everything I've learned in the last few days has really emphasized connectivity, systems thinking, systems approaches. That's what the Seascape approach is about. Our government decision-making doesn't necessarily work in that way, I think it's fair to say. It might perpetuate silos. I've had a few conversations with people about licensing and about how they get bounced around between different departments. Nobody really knows whose responsibility it is, etc. So I think there's a bit of a challenge for us there in terms of how do we embed something that is inherently a systems-based approach into a system that doesn't operate in that way necessarily. And a question for you as well, do we have a unified ask that we feel like we could collectively get behind that would make seascape restoration easier for you all, even if it's step one on a longer path? Lots of talks have mentioned the importance of policy. Quite often it's the last thing people mention in their slide deck, right at the very end. And we communicated this to policymakers. I'm like, yes, but what did you say? What did they say? What was the result? So do we have something we feel like everybody here could unite behind and get behind, maybe campaign behind, that would really make a difference to the work that you're trying to do? Thank you very much.
Thank you very much, Georgina. Whilst you're doing that, we do have a few moments for questions. So many of us can multitask. So if you can, any questions for Georgina?
Hello, my name's Sahara. I'm from the Society for Ecological Restoration. I will pounce on you tomorrow. We need to talk. So I'm sat here next to RJ, Alison, and Jo was here, but she's not anymore. And we're all leading networks or alliances that are bridging policy and practice. And I want to know what we can do to support these very, very, very busy people to do their job better, because obviously we might be in a position to help there, and either in training in how to communicate better to policymakers, or what would your ideas be for that?
Yeah, so I think there are a few things. I think definitely influencing and advocacy is an art, not a science. So there's definitely a lot of skills that people probably do on a day-to-day basis that they don't realize they do, but there are definitely systems and processes and ways of thinking that can help. So I think running workshops is a really good thing to do. I think it is quite difficult to build policy advocacy capacity into funding proposals, and quite often it gets missed off. So all of the amazing work you're doing, I want to take that and go and talk to people about it and do more targeted government engagement with it. But if you don't have somebody who's dedicated in doing that, quite often the sort of policy and government engagement work gets left to the person that's also trying to manage the project, run the project, do the fundraising, do the implementation, etc., etc. And it is a very specific set of skills to be able to do it effectively. I hope I don't mind saying, otherwise I wouldn't have a job. I think make sure you build that capacity in, whether it's even just a workshop with government and decision makers, then find a policy or a government affairs colleague to kind of come and run that workshop for you, or to help you design it. So I think those are a few things that we can do. But I quite often find policy gets missed out of as a kind of core skill that's built into projects. So please make sure it's built into projects.
I second that.
Yeah.
Great. Thank you very much, Georgina. So there we are. Thank you very much to the 4 speakers in this session, a range of topics, all interlinked, of course. So if we give them all a round of applause, thank you. And then our mystery slide, is Leah here? Okay, are you going to say something?
Hi, my name is Leah. I'm a PhD student at the University of Oxford in the Seascape Ecology Lab with Lisa Wedding. My PhD in the broadest sense is combining maritime heritage with marine science. I'm sorry for yet another QR code, but essentially this is a survey. This is for anyone who would consider themselves a user group within marine spatial planning. So it doesn't matter if you're an academic looking at restoration initiatives. It doesn't matter if you're in policy, if you're looking at national parks, if you're an extractive user. This is dedicated to you. You do not need to know anything about heritage. It's just your opinions. And anyone who's willing to fill this out, I would be very much appreciative.